mean, thanks for all the positive words that Laura gave us in the previous talk. We've worked with York for quite a number of years, and it's a real pleasure to see all the advanced work that Laura has done. But of course, to do that work, you need a good imaging workhorse, something that you can come to day in, day out, doesn't need any special calibration, will look after your cells in a really good format and keep them live for a long time. And for Laura's particular application, images that you can easily segment and give these tracked segmentation output. And that's what we have with the LiveCyte. So you see this on the screen here. It's a boxed microscope system. This chamber is your 37-degree chamber. You can see a motorized stage in there and the objectives down there. Where you would normally have a phase contrast head, we've got our quantitative phase tychography head, and this gives us these beautifully segmentable images that are completely non-perturbing to your live cells as you go on. And we just work with conventional imaging plasticware. So it's a very, very simple system to use. And we already combine it with a lot of software that presents the output. So I'll continue with this introduction just through to the first biological example, and then Meetal will take over and give us a real deep dive into what has been the bread and butter workhorse of what people are doing on the LiveCyte. And my email's up here if anyone has any questions that we can't squeeze in at the end, because I appreciate there's a lot for me and Meetal to try and squeeze into 30 minutes. But thank you all for attending. So I need to get my screen to move on. So it's a long-term time-lapse imaging and analysis system. So even without Laura's extra layers, we already do analysis of thousands of live cells. We work up to 96 well plate format. We have our quantitative phase imaging system, which all of Laura's images were based on, but we can work with up to 7 fluorescence channels, which makes it a really good long-term live cell imaging workhorse. And you know what we've really put a lot of effort into is drilling right down to the single cell level and understanding this strange cell behavior, these partial populations or single cells within a well that may be different to the rest through time as they differentiate or react to treatments. So just again to review for those of you that don't know, this is a quantitative phase image. This is just cells. They look like they're probably fluorescently labeled. They certainly don't look like what you would expect from a normal phase contrast image. In fact, they look almost exactly like a diffuse CFSE stained image. This is what gives us the images that are really easy to segment, really easy to derive all sorts of phenotypic parameters from. But the beauty of this is it's completely non-invasive, no labels, no special preps, the cells stay alive. If you image CFSE, it's one of the most phototoxic compounds on the planet. Great for flow cytometry, but your cells will be dead within hours. And when we run a label-free assay, all of these assays drop out automatically. So before you even consider the particular experiment you might have designed, you get proliferation information, you get growth information, you get morphology information, you get motility, mitosis, and single cell lineage. So if we look at these in a bit more detail, this is all from one label-free image. We can measure confluence just like you might with, say, something like the IncuCyte. But completely unlabeled, we can give you a true cell count. So we're now coming into the area that's a bit more like you would get with endpoint assays with a high content imaging system that you're more familiar with from that side of the fence. But our content really comes from the time-lapse single cell data. So we're still a high content imaging system, but we're not the super high throughput multiple plates at a time because our throughput is the time lapse. And when you can distinguish cell count from confluence, then you know whether your cells are becoming megacytic or whether they're truly dividing. And if you measure when they're truly dividing, you get information like cell doubling time. You get the total mitotic events. You get mitotic index and mitotic time. And when you've put all that together, you can start looking at how cells grow. And one of the really interesting things about quantitative phase imaging is the density of the image, rather than being of a fluorescence label, it directly equates to the dry mass of the cell. So we're measuring true cell dry mass. This gives us an extra parameter when we're tracking cell movement as well, because we know that mass won't change from frame to frame too much until the cells divide. So as well as measuring cell mass, it gives us this extra little handle on tracking. We measure the morphology, and we measure the shape and other parameters. We measure the motility, and what's really nice to think about these graphs, all of these bee swarm plots and all of these individual tracks, these are all made of dots of individual cells where we have that data you can drill right down to. The software has really been built for that single cell understanding. And if you put all this together, the motility, the growth, the cell division, well, you know what parent cells were, you know when they divided, and you can track cell lineage. And Meetal will dive into some of these in a lot more detail shortly. But every LiveCyte assay gives you all of this data, a lot more than you might expect. So here's just an example image, just by way of showing you some nice videos, but I think Laura's already shown some nice videos, so I'll not dwell on this, but here we were imaging and segmenting cells. So from this you get more metrics that matter, greater physiological relevance, and deeper correlated insights into your data. Now, I'll give the first of these. I'll just talk about our advanced T-cell killing assay briefly, and then Meetal will dive into the other applications. I wanted to give the T-cell assay just because I've been quite close to the T-cell community for a number of years, and it's one of our more recently released assays, and I think it really shows off the system nicely. So T cells, primary cells, can often, if you're looking in a therapeutic environment, already have been engineered. You don't really want to perturb them. So it was really precious for us to keep the T cells unlabeled. But to make an advanced T cell killing assay, this is something that gives you just more than cell death, which a conventional T cell killing assay, we do still need to use the fluorescence capabilities of the system. And with that, we can measure an awful lot of data, and that's pictogrammed here on this plot. So as well as cell death, we're looking at when C cells interact and how long they interact with the target cells, the number of T cell contacts that lead to death, the final T cell contact time, so what the total number of time of contact was. Because it's a killing assay, we measure the cell deaths, but we don't just measure cell deaths. Because we can detect target cells, we can look at the target cell numbers. So we can check that you had good seeding density at the start. You can also see if the targets are proliferating before they die or stabilizing, and that's quite interesting. We've got data that show even non-targeting T cells at high density will reduce proliferation of target cells, and T cells attached at death. And this is all done because we detect cell death with a fluorescent marker, and then we backtrack through the series of time-lapse images to see all of these events that happen. And we can really distinguish small differences in T-cell behavior with this assay. So here's a typical movie of what's going on. The T cells are the small white dots, and here you can see the segmented target cells. And the beauty of segmenting the target cells is you can see when T cells touch them, overlap them, and in this case, they're one curled up and died. If we zoom in a little bit more detail and play through this movie, then you'll see the T cells coming in contact with the target cells. And towards the end of the movie, it just gets busier as the cytokine concentrations build up towards the target cell and cell death happens. So we measure all that through the time lapse. And that's all presented in dashboards, and Meetal will talk more about dashboards. But all of these graphs and plots, and you saw this on the very first example slide at the beginning of the presentation, they're all there to really present the data in a user-friendly, simple, and digestible format. With that, I'm going to hand over to Meetal, who will go through more, and then hopefully we'll still have a little bit of time at the end for questions. Thank you very much, everybody.
Thanks, Pete. Let me share my screen. Okay, so as Pete mentioned, some of the applications in addition to T-cell killing, I think he described them pretty well as a bread and butter experiments, and I think cell growth and proliferation is definitely described as that. It's pretty much something that's done in every lab, and that's mostly looking at this increase in cell count. But the slightly less well measured output is increase in biomass. So this indication of growth. And conventionally, as a proliferation assay, you might look at something like confluence versus time. So just looking at the area of the image that's covered by cells. And normally that's as a percentage of the total area, and can be plotted on a graph as a proxy measurement of proliferation. And although confluence assays are really quick and easy to do, they do have some drawbacks. So one of those is that it can't separate growth from proliferation. It can't also look for any sort of differences in cell size or morphology that your treatments might be impacting cells. And where LiveCyte comes in is it can actually measure growth and proliferation independently. So you've seen some of the high contrast images that Pete and Laura showed, and from that we can quite easily measure label-free cell count. But what we can also do is measure the growth and the mass of the cell using our unique metric dry mass. And I just wanted to go through a really quick example experiment that we did in collaboration with the University of York. And this was initially looking at measuring fluorescence toxicity over a long period of time. And essentially, this was trying to look at some of the subtle behavioral changes, specifically growth and proliferation in response to cell-DNA fluorescence stain. So we had cells which were labeled and also cells which are unlabeled, and these were imaged over a 5-day period on LiveCyte. In addition, we also wanted to look at several different illuminations to see how that might also be impacting cell behavior. And what we found by looking at the time-lapse images initially is cells look pretty similar at the beginning of the experiment. It's not much difference. But if we fast forward to the end of the experiment, we can see that after 5 days, there's some clear differences. One of them being the cell-DNA cells definitely look like there's much less cells within that field of view. But the other much more stark kind of morphology change is that cells look a lot larger. A lot of them actually have are binucleated. And this was something that we wanted to be able to measure separately. So initially, because we were seeing changes in the proliferation and also the mass of the cells, we couldn't really see any clear trends from the confluence graph. There was just too much going on. What we really needed was to be able to measure the growth and the proliferation of the cells independently through looking at cell count and dry mass. So looking at the cell proliferation from cell count and the cell doubling time, we were actually quite easily able to pick out trends in the cell doubling time for the labeled cells in particular, and we could see that with the higher illumination cells, they were definitely slower to proliferate, and this was definitely dependent on the illumination. And if you compare that to the unlabeled cells, there was really no change in the proliferation of those cells. And we were also able to look at the individual cell growth, and this showed what we were seeing by eye, which was that those label cells were becoming heavier over time. So they seem to be accumulating mass, and this might be because they weren't able to divide fully, or there might have been changes in the cell cycle. which meant that there was a lot of binucleated cells that weren't able to fully divide, and that was the change of mass that we were seeing. And again, this was dependent on the dose of illumination. So again, the turquoise line was the highest. And as we're able to also gain these really high contrast images, we're not just able to look at proliferation and growth on a population level, but we can also look at division a bit more intimately by looking at mitosis. So just looking at these A549 cells, you might be able to see the point at which the cells divide, they actually ball up. And this balled up phenotype is something that we can actually measure quite well. And that gives us information on things like the number of mitotic events, and how long those cells actually spend in mitosis before they finally divide into two cells. What it also gives us is single cell information on the cell lineage. So this is something that's very much a single cell exclusive. So it's something that we can do because we're able to segment and track and understand mitotic events in more detail. And what it means is that you can delve into a particular population of cells within your well plate, and you can see sort of where the parent and daughter cells are, and you can assign each of those a lineage ID. So you know exactly which daughter cell came from which parent cell, and the point at which that happened. And on our software, you can actually see that as a lineage tree. So you can see multiple lineage trees here, which are each subpopulation of cells. And you can compare and contrast these. So you can see the point at which they divided, what was the dry mass of that cell, and you can compare it to other cells that also divided. And that allows you to look at things like aberrant divisions, divisions which you might have included cells which were daughter cells which had different dry masses, and kind of incomplete divisions and things like that as well. So it allows you to look at heterogeneity in your population as well. So next I'm going to move on to cell motility and migration, and LiveCyte does this really well because we're able to segment and track every single cell. And principally, the kind of assays that people do on LiveCyte are random motility assays and single cell scratch assays. So firstly, talking about conventional wound closure metrics. So typically you might do a scratch assay if you're looking at an in vitro model of wound healing or tissue repair, and you scratch a well and look at how the cells move into the wound. And essentially you're looking at something like the area of that wound over time as the cells close in. But what does that gap closure rate really tell you? One of those things, yes, is migration. Yes, you can tell that, yes, cells are going into the wound a little bit faster or slower. But it's very much a proxy measurement, and it's sensitive to a lot of changes. One of those being your initial gap area. So especially if you're manually scratching your wells in a plate, you might find that you're going to have variability in your scratches, and this can affect your final results. Things like seeding density and proliferation can also affect how your cells move into the wound. If you've got a lot of cells, that you might find they're actually just pushing into the wound. And even with serum-free media, you might still see a level of proliferation over time. Other things that might affect it are things like cell area and morphology changes as well, if you're adding particular treatments. So really what we need is something to be able to measure scratch assays, but also look at metrics which are independent of the wound area. So something that's able to look at single cells and how they're reacting to the scratch. And that's exactly what LiveCyte does. So you can see a screenshot of the dashboard. So Pete mentioned for each of our applications, we also include a dashboard within the software, and that has all the metrics that are compiled for all of your treatments and wells in a really easy to use way. And what we include is things like the conventional metrics. So you can see you've got your T.5, so looking at kind of the point at which 50% of your wound area is closed, and also wound closure rate. But we've also got other metrics. So we can look at the track speed of the leading edge cells, so the cells that are right at the edge of the wound which are moving in, and we can see their directionality. So how much preference do they have to move into the wound and not maybe sideways or back on themselves. And we can also look at kind of things like collective migration as well. So this really gives you an idea of kind of a wound area profile that's independent of just kind of are your cells moving into the wound. In addition to that, you also get a variety of other applications as part of your assay, and that includes things like proliferation and morphology. So if your cells are proliferating or if they are changing in their morphology, then that will also be tracked over time. And essentially, these extra metrics just allow you to understand not just what treatments or wells which are closing faster, but it allows you to understand how those cells are closing and what might be the reason. However, in some cases, you might find that a collective migration assay might not be the right assay for you, and you might need to look at individual cell migration. And in those cases, you really need to look at random motility assay. However, to do that, you need to be able to track your cells throughout the experiment, and that can be pretty time consuming and laborsome. However, with LiveCyte, you can do that automatically. It happens in every assay, regardless of if you're doing a motility assay. So you will always obtain that information. And the cells are all tracked very reproducibly, which alleviates any sort of variation that you would have if you were manually tracking your cells. And again, you get all of these metrics compiled within a dashboard. So these are things like the velocity of your cells and the average track speed, all throughout the experiment, as well as sort of how those cells are moving. So are they have they got a particular directionality, and are they quite confined, or are they moving quite far away from their location? And ultimately, that just means that you can choose the most suitable motility assay for your cells, whether that's a random motility assay or a scratch assay. Another part of the LiveCyte software is that we can basically isolate and look at specific subpopulations in a bit more detail. So this becomes really useful when we're looking at co-cultures and heterogeneity. And we're able to do that through our single cell tracking. So that enables us to sort the cells based on potentially fluorescence or even morphological phenotypes. So here, for example, we're able to capture just the cells which are fluorescent within that population of cells, and then be able to do analysis on those separately. So we could also incorporate that into the dashboards, for example, and look at cells which are in and out of the gate, or you can also export that to do your own analysis. And an example of where that was quite useful is particularly where you're looking at co-cultures. You might want to see how those cells react in a co-culture compared to when they're just in isolation. And we had a look at several progenitor and differentiated cells to see if there was any particular difference in markers. We saw some quite similar patterns in terms of track speed. So it seemed like whether the cells were in isolation or co-culture, they clearly had a similar pattern in that differentiated cells were slower than the progenitor cells. But we also saw that the differentiated cells seemed to proliferate a lot faster when they were in co-culture with progenitor cells than when they were isolated. So it definitely seemed like some sort of signaling mechanism was causing that. And it's not just confined to fluorescence versus non-fluorescent cells. So we can use the same label-free cell subtyping to use it on kind of morphological differences. And you can see here with the primary prostate cancer cells, the users here didn't really want to use fluorescence because it would have perturbed their primary cells. So because the two cell types you can see are quite different in their morphology, they could actually use label-free metrics to do exactly the same job. And finally, we'll wrap up.
We're quite tight on time, so it might be worth skipping over Neurite just to the last slide so people get a chance to ask some questions, I'm thinking.
Yeah, so just quickly going through one of our newer applications. Essentially, you can see from the image, you've got essentially we can measure network analysis through our label-free QPI imaging. So instead of having to use fluorescence, which might perturb your cells, you can actually analyze your network automatically with just label-free imaging, and gain lots of different metrics, which I can actually show you on the next slide. So again, everything's compiled onto a neural outgrowth dashboard, and this allows you to look at kind of branching complexity and the total length of your network, and various other things which can all be exported to. So I'll leave it there. If you've got any questions, then feel free to ask at this point.